Okay, we are moving on to gluconeogenesis in chemistry. Here we go. Gluconeogenesis, a pathway for de novo synthesis of glucose from non-carb substrates such as pyruvate, lactate, RBCs, and muscle. Just making sure the camera's in a good enough spot to make this work. Okay. During anaerobic exercise, glycerol from adipose and glucogenic amino acids from muscle protein using both mitochondrial and cytosolic enzymes found only in the liver, kidney, and intestinal epithelium. Gluconeogenesis mainly occurs in the liver during prolonged starvation. The kidneys also become major glucose-producing organs. The brain preferentially uses glucose, but can, in states where glucose is not available, use ketones. It serves to provide glucose, especially for the brain and RBCs, which require glucose for energy. Gluconeogenesis occurs during fasting, starvation, or intense exercise, and is highly endergonic. Remember that humans cannot make glucose from acetyl-CoA. Purpose of gluconeogenesis: to maintain blood glucose levels in the fasting state by reversing glycolysis. All of the 10 steps of glycolysis are reversible except step 3. The 3 irreversible glycolytic enzymes are the enzymes used in steps 1, 3, and 10. Glucokinase hexokinase replaced by G6-phosphatase. Phosphofructokinase replaced by fructose 1,6-bisphosphatase. Pyruvate kinase replaced by pyruvate carboxylase and pep carboxykinase. 4 replacement enzymes are needed to replace the 3 irreversible glycolytic enzymes. Most of gluconeogenesis takes place in the cytosol of the liver cells, and to a smaller extent the kidney and small intestinal cells. Gluconeogenesis uses lactate and amino acid as substrates. Lactic acid, formed during anaerobic respiration in skeletal muscle, is a common substrate for gluconeogenesis. Lactic acid may also come from RBCs, which obtain energy solely from glycolysis, as they have no membrane-bound organelles for aerobic respiration. Lactate is transported back to the liver, where it is converted to pyruvate by the Cori cycle, using the enzyme lactate dehydrogenase. Alanine is a glucogenic amino acid derived from glucose-alanine cycle. Both lactate and alanine will be converted first to pyruvate before entering gluconeogenesis pathway. The Cori cycle is an energy-sapping cycle and comes at the cost of a net loss of 4 ATP. This cycle prevents lactic acid from building up, lactic acidosis, in skeletal muscle. The first step in gluconeogenesis occurs in the mitochondrion, and the rest is in the cytosol. Step 10, reversal. The replacement for pyruvate kinase is a 2-step process. The first step converts pyruvate to oxaloacetate, which occurs in the mitochondrion. Pyruvate carboxylase is the replacement enzyme. Biotin is an essential coenzyme for this step. Biotin is B7. Biotin deficiency will result causing pyruvate to build up. This results in excess lactic acid production. Oxaloacetate passes into the cytoplasm. ATP and acetyl-CoA are allosteric activators. The second step in reversal takes oxaloacetate to phosphoenolpyruvate. This occurs in the cytoplasm. This is catalyzed by PEP carboxykinase. Lots of energy is required for this step. Energy is provided in two ways. Decarboxylation is a favorable reaction. GTP is hydrolyzed. PEP carboxykinase is inhibited by insulin. PEP carboxykinase is facilitated by glucagon and cortisol. Step 3, reversal. PFK, or phosphofructokinase, converts fructose 6-phosphate to fructose 1,6-bisphosphate. The replacement enzyme for PFK is fructose 1,6-bisphosphatase. This is a rate-limiting step for gluconeogenesis. It is stimulated by ATP and citrate. It is inhibited by AMP and fructose 2,6-bisphosphate. Step 1, reversal. Glucokinase converts glucose to glucose 6-phosphate. Glucose 6-phosphatase converts glucose 6-phosphate to glucose. It is present in the ER of the liver and kidney cells, making gluconeogenesis possible. G6P is hydrolyzed as it passes into the ER. Gluconeogenesis does not occur in the muscle and the brain. Vesicles filled with glucose migrate to the plasma membrane, fuse with it, and release glucose into the bloodstream. Gluconeogenesis is inhibited by insulin and stimulated by glucagon. Energy usage. Gluconeogenesis is an energy-consuming process. 6 ATPs are used, producing 1 new glucose molecule. Acetyl-CoA is not used because it requires more energy to make glucose from acetyl-CoA than from lactate or alanine. Fate of new glucose. If the new glucose is not used immediately, it is converted and stored as glycogen in the liver or in skeletal muscle. Here's the gluconeogenesis pathway, which has arrows going both ways. So I'm not going to read through it, but there it is in video. The next section is on Krebs, and there's a lot to it, so wait for the next video.